Hey, how you doing? Hi. Good. Hey, Lisa. How are you? Good. Hello, how are you doing? I'm Karen Savage. I'm introducing you to Penny.
Oh, okay.
She's out.
She's out till next week. Sure. I gotta go back to this.
Her son is really excited about
That
it.
was on the last question.
So we usually do this afterwards, kind of discussion.
No, that was a big issue.
No walls come down. I had a
It's just
It's just the 45s that you have to go and get that from outside.
Electricity. Hey, good afternoon, everyone. I am Karen Savage. I'm here today for Penny Jester to introduce today's speaker. We've got Dr. Jeff Edberg. He is the director of the CCTS Core Lab and Biorepository, and he is going to tell us all about what he does and how to build a repository from specimen collection to usage. Welcome, Dr. Edberg. Thanks for being here.
So I'd like to keep this informal because I know there's probably a lot of different backgrounds here and people are coming with different perspectives and different levels of questions. So please, if you want to talk about something, just stop me and we can talk in more detail about any of the things in the presentation. So I'd like to talk about biorepositories, obviously, but I think before one even builds a biorepository, it's really worth stepping back and asking, what are you trying to accomplish? What are you actually doing? What are you storing? Why are you doing it? Because at the end of the day, if you store a bunch of stuff, whatever that stuff is, and it turns out not to be useful for any questions anyone wants to ask, then that was kind of worth the time. So it's really worth spending a lot of time thinking ahead of time, what is it that I'm really trying to do, and what am I trying to accomplish? Are we in the slideshow?
Supposedly. Sorry guys, I'm doing this get tech support, I guess. I've done presentations here.
Yeah, no,
Any trouble?
It doesn't look like you're in PowerPoint.
Mm-hmm.
Yeah.
Okay.
So if you go down to PowerPoint,
Oh. And then slideshow.
From beginning, I guess.
All right, now let's get started.
There we go. So maybe the first question, so what is a biorepository or a biobank? Well, it's really a library in which specimens are stored for clinical and or research purposes. And it's important to keep the distinction between clinical uses and research purposes because there's a lot of different regulations that get invoked if you start to talk clinical applications for any of the specimens that are stored in a biobank. So if samples are intended to be used for clinical use, that really means that they're going to be used in a clinical application. So information derived from that specimen is going to be used to make a decision about patient care, or that information is going to in any way touch the medical record. Then you have to follow a very different set of rules about how those specimens are collected and stored and what information is available on them. And the laboratory and the staff have to have appropriate certifications in order to work with those specimens before they can be used clinically. And that typically would be a CLIA or CAP type certification to function in the clinical setting for these specimens to be used. I'm going to focus exclusively today on the research use of specimens. That's really then specimens stored to be available for research purposes. This is not intended to be used to make medical decisions or to be put into the medical record in any way, shape, or form. Now, that doesn't mean then that people don't have to be careful and follow the same rigorous guidelines that one will follow when samples are being used clinically. But it doesn't mean that you have to have those regulations and certifications in place in the lab in order to do the research. And as I'll talk in a little bit, there still have to be good practices followed in the lab. You still have to follow best practices for what it is you're doing and understand how this sample is going to be used in order to make sure that they're useful. So the other issue that we need to keep in mind is that just because you have a blood or a blood derivative, whether it's plasma, serum, whatever, that's fine. It goes into the freezer, but by itself, it's not really very useful, right? You really need to know something about that sample. And so in the past, people said biospecimens can have annotated data, but I think really going forward is they should or they must have annotated data associated with them. And that data can be a variety of different sources. It could be simple demographic data. Obviously, you want to know where is this sample from? Is it from a male, female? Sometimes ethnicity is important, age is important. Certainly, there can be research-based results that one might want to have associated with the specimen in order to make it useful for further use. But there can also be sometimes clinical data from the patient whom that biospecimen is collected that might help inform how that specimen can be used and how the results are interpreted based on the types of results you get. So before we get into it, I'd just like to review 3 different types of specimens and what the UAB resources are for working on these types of specimens. So obviously in all of this, I won't make a distinction between a biospecimen from a healthy individual or a patient with a particular phenotype or disease. They're biospecimens, and obviously the type of information associated will vary depending on whether it's a healthy person or a person with an autoimmune disease or whatever. But I think the basic guidelines for building a biobank are really the same whether it's healthy or disease. If you're interested in tissues, and that could be affected or unaffected tissues, there's a very good program here on campus in the Department of Pathology. It's a tissue procurement and shared facility over in Ziegler that Dr. Grizzle runs. He can get, with appropriate IRB approval and consent processes, he can get tissues from different patient populations and unaffected tissue for investigators who've had a very successful program for many years doing that. I'm not going to talk at all today about tissues. The issues related to the storage and collection of tissues are quite complicated and quite distinct from those involved with working with blood, which is what we focus on mostly in the CCTS. Certainly, if we're working with blood or blood derivatives or urine, I would suggest that our lab in the CCTS, which I'm the director of, and I'll give some contact information at the end, would be a place to get information about how to do this. Doesn't mean that we have to do it for you, but certainly you're welcome to use this as a source of information and guidance on how to think about collecting and storing these types of specimens. And then lastly, there's a lot of interest in the role that the microbiome plays in health and disease. There's a lot of interest in collecting stool for microbiome analysis, and again, through the CCTS in collaboration with other centers on campus, Dr. Casey Morrow in cell biology leads an initiative where they've worked out all the details of specimen collection, shipping to the lab, and then working those specimens up for microbiome analysis. And it's a very nice program where he integrates both the collection, the analysis, and the actual data analysis together to help investigators look at the microbiome. So before we build the biorepository, what should we think about? Well, you really need to think about, well, how are these going to be used? And what do I need to do to get the right specimen? So that really involves how am I going to collect, where am I going to collect, what am I going to collect, how are the specimens handled and processed, how are they stored, and how are they retrieved? And a lot of these things may seem very elementary or very straightforward, but you really have to give some thought to these things because it really ultimately depends on how do you think these are going to be used. Certainly, you're collecting for a purpose, so there must be a study you're doing, and there's going to be a particular need that you're trying to address with those specimens. And you need to make sure that your collection and storage and processing procedures are compatible with what it is you're measuring. So, for example, if you're measuring certain cytokines in blood, which are inflammatory molecules that help regulate the immune response, some of those are very labile, and they don't do well if the sample is stored at room temperature for even a couple hours. And so those samples have to be worked immediately. Some analytes are sensitive to light. So you need to understand what it is you're trying to measure so you know how to handle your specimen. You also need to give some thought to how these samples might be used in the future. Certainly for your current study, you want to measure certain things now, but there are probably additional uses that you might think of in your disease or situation that you might want to do in the future. You need to make sure that you're collecting materials that are compatible with those potential downstream applications. And then certainly specimens can be used as a mechanism to leverage collaborations and other studies in the future, assuming that appropriate informed consent is obtained, of course. So you need to think these things through before you really do anything with your biobank and biorepository. So I mentioned informed consent. I'm not going to say a lot about this other than that you, again, need to think about this, right? So your consent form needs to consider how are these samples going to be used in the research. So what's the participant consenting for the study? What does that actual language say that the sample is going to be used for? Other things you need to think about, will research findings be shared with participants? In most of the studies I work in, we typically don't share findings directly with participants, and we say that explicitly in the consent form. Another thing you need to think about is what about incidental findings of potential clinical importance? How's that going to be handled? This actually does come up. We've had a situation. I study autoimmune disease in patients with lupus in particular, and we were doing a genetic study, and one of our collaborators was working on genes that are on the X chromosome, and it turned out we found a patient who had Turner syndrome. It's a very young patient, and that wasn't known at the time. So what do you do about that? So the sample was not acquired in order to provide any clinically relevant information, but nonetheless, something important came up, and so we had to go to the IRB and ask how to deal with that, and it was more complicated because the found the patient was a collaborator at different institutions, so there were multiple IRBs. We worked it out eventually where the information was given to the clinician to then approach the patient and verify the diagnosis. But you need to think about these things when you're doing particularly genetic assays and other assays too. Are there going to be potential incidental findings that you might need to do something about? Certainly your consent form will need to articulate how your specimens will be used in research studies. And that you need to work on the language there very carefully to ensure that you give yourself enough latitude that you certainly know what you're going to do with step 1, but what about steps 2, 3, and 4? As your research builds on results, have you structured your consent form to allow you to continue your research to use those specimens in further studies? And again, this issue of facilitating future studies, that's something that's been particularly useful in our work in autoimmune diseases. We've been very fortunate to collect a very large biobank of patients, specimens from lupus from multiple institutions, and with appropriate consent, we've been able to form a number of collaborations over the years that have led to many, many more studies that have come out using our specimens and working with us directly to utilize those materials to further our understanding of the genetics of lupus. So it's really important to think about your informed consent process and to ensure that you're writing it so that it's fair to the patient, that you're upfront in what you want to do with the samples, and what might happen in the future. In our consent forms, we always build in a tiered consent process where they can say you can use my samples in this study, and that's it. Or you can use my samples now, certainly, but if you want to do something else with them, that's fine, but contact me first and see if I'm interested. Third is you can use my studies in future, or my specimens in future studies, as long as you don't identify anything about me or who I am. I believe it's not a limited consent, but it allows us then to use those samples in future studies. And it gives the patient the control of how their samples are used in the future. Okay. So, again, obvious things, but something you need to think about. So the specimen needs need to dictate the nature of your collection, right? So you need to make sure your informed consent is done appropriately. Yes. Sorry, I kept forgetting. So let's talk about the life cycle of research specimens. So we talked a little bit about thinking about specimen collection and how they're going to be used. So we really need to talk about then how do you implement this lifespan. Once you understand what it is you're collecting, how you think you're going to use it, what is it that you need to do to build a good biobank to allow your specimens to be useful for your research project and to facilitate future work. And there are really multiple steps in this process. And we'll talk a little bit today about specimen labeling, how everything is labeled, how are they transported, how do they get to where they're supposed to go, how do they get processed, and then how do they get stored. And then issues related to specimen use. For most blood-based specimens, the thing you need to keep in mind are those freeze-thaw cycles. So you put something in the freezer, that's great. You take it out, you thaw it, you use it, you put it back in the freezer. That freeze-thaw is not a the specimen will change in response to that freeze-thaw. So sometimes it doesn't matter if you're measuring antibodies in the serum, they can be freeze-thawed a number of times. It probably doesn't make that much of a difference. But some things are actually very sensitive to freeze-thaw. So you just need to think that through. That actually goes back to the specimen processing step then. So how big your allocrops need to be. You need to think that through ahead of time to understand what it is that you need to do to have specimens that you can use. So when we think about specimen collection, what kind of tubes do we use? So this is from Beck and Dickinson. This is on the website. You can just Google BD vacuum painters. There's a lot of different kinds of vacuum painters, and these aren't all of them. But you need to know what it is you're going to do your samples in order to know which one to use here. So the type of blood collection tube you use really depends entirely on what it is you need to do with your sample. So if you're interested in cells, for example, you're not going to collect a serum tube, right? The serum's going to clot, and then all those cells are going to be locked up in that blood clot. And all you're going to have left then is the liquid part of the serum. So cells can be very useful if you use a serum tube. And we go back to our diagram here. Typically, these tiger tops or red tops here are your serum tubes. They clot in, they're anticoagulant, and you're not going to do much with white blood cells there. If you're interested in cells, you need to have an anticoagulated sample, and that would be any of these down here. Typically, green, purple, and yellow are the most commonly used. And I'll mention a couple examples on when you might want to think about one over another. If you're interested in plasma serum, you need to understand the difference between plasma and serum and know which tube to use. If you're interested in nucleic acid, which is DNA, RNA, you need to know whether you're interested in DNA that's associated with the cell, which is the traditional application, so DNA from the nucleus or the mitochondria, or are you interested in circulating nucleic acid? And there are some newer diagnostic tests and ideas coming out about circulating RNAs that might be very useful for diagnosing certain types of cancer. So let's go through each of these individuals. So blood cells, if you're interested in blood cells, you're generally going to need anticoagulated blood. But the anticoagulant, which is the color of the tube, can make a big difference in the types of assays you want to do. So for example, the purple tops, which are EDTA tubes, if you want to do B-cell work, if you're an immunologist, B-cells don't like EDTA, and they're not going to be very viable after isolation. So you really need to avoid UDPA tubes, and typically if we're interested in B cells, we use green tops or heparin tubes. Time and temperature is important, right? So the longer the cells sit after draw, the less likely it's going to be viable once you isolate those cells. Temperature is important. Again, a lot of my work has been in B cells. When B cells are put on ice, or blood is put on ice, and you isolate B cells from that blood, they typically don't work really well at that point. So we tend to avoid icing or refrigerating samples if we know we're going to be interested in B cells. That's not true for all cells. So it really depends on understanding the cell type you're interested in and what it is you think you want to do with the cells that will dictate how your sample is handled after collection. And obviously, whenever you're working with cells, it's really the timely processing and isolation that's really going to be the most important thing. As soon as you can get those cells isolated out, the better you're going to be. Oftentimes, if you're interested in white blood cells, people use those CPT tubes from Dr. Dickinson. These have a matrix in them that allow you to separate out the peripheral blood mononuclear cells, or PBMC, in one centrifugation step. They work very well. They're a little expensive, but they work, and they allow you to draw the blood and put it right in the centrifuge and get PBMCs in about a half hour or so. There are manual ways to do it, which have some advantages and disadvantages, but the point again is just time, right? You want to work fast. So work with cells, you need to get these going quick. So plasma serum. So we need to make sure we understand the difference. Remember, serum, that's the liquid part that separates out after blood clots. This is typically in orange top tubes or tiger top tubes. Sometimes those tubes have a little gel barrier matrix in it. Those are really good because it helps get a really clean separation between that clot and the liquid part, the serum. The disadvantage is that sometimes there are some analytes that might be sensitive to that gel material and may not be compatible with that. So then you have to use a plain serum collection tube. Plasma is also the liquid part of blood, but that remains after the blood is collected in an anticoagulated, so the blood is not clotted here. You can spin down the red and white blood cells, and the liquid part that's left on top is the plasma. So serum is after clot, plasma is when it's anticoagulated. Again, as with cells, time and temperature is important. Some analytes in serum and plasma are very sensitive to time and temperature, and you need to understand what it is you're going to do with your sample about how to deal with those issues. So, nucleic acids, the last type of biomolecule I'll talk about. This is typically DNA and RNA. So remember, DNA is deoxyribonucleic acid. That's the genetic material in your cell, typically the nucleus or the mitochondria, and it carries the genetic information for all the cells in the body. 90% of the time, that's where you're getting DNA from. There can also be circulating DNA that sometimes can be harvested. And there are ways to do that from serum or plasma. And again, there are some issues there. When blood clots, there are a lot of enzymes that are activating some DNA degradation. So typically, anticoagulated blood is better for that. RNA is ribonucleic acid that's present in all cells, and it's what carries the instructions from DNA and controls the synthesis of proteins. RNA is very susceptible to degradation. There are proteins called RNases that degrade RNA. They're everywhere. And they will degrade your RNA very quickly if they're not handled appropriately. So if you're interested in RNA, you really need to give thought to special handling conditions. There is a commercial solution called patch gene tubes, blood collection tubes. These are tubes that are pretty tall, but they have a lot of liquid in them. They collect about 2 or 3 mL of blood. And the liquid part is their proprietary reagent that lyses the cells and stabilizes the RNA so it doesn't degrade very much. And once blood is drawn into these tubes, it's very stable, even at room temperature for days, and then in the freezer for months to years, depending on how you freeze it. So it's a good way of quickly stabilizing your sample. But the issue there is, where do you need your RNA to come from? You need RNA from a particular cell subset. So if I'm interested in RNA in T cells, for example, a patch tube is not going to help you because a patch tube is going to lyse your cell, everything in blood, and you can lose your cells at that point. You're pooling all your RNA together. So if you're interested in RNA from cell subsets, and it's important to remember that blood is not a single tissue, right? Blood is made up of a lot of different types of cells that have different functions, different proteins, different RNAs are expressed in those cells, and sometimes looking at whole blood is fine, but depending on what you're doing, I'm an immunologist, so when I look at blood, that's not a single thing. That's a lot of different types of cells, and so I want to separate those out and do different things to them. So if I want to get RNA from those, which we sometimes do, time is important, and you need to work those cells up as quickly as possible and get them lysed into something that will stabilize that RNA. DNA tends to be much more stable. For a number of studies, we actually have multi-site collaborations where people are sending us patient samples. They have Friday clinics, so they send on Friday, we get it on Monday. They just ship it room temperature. We can get the blood on Monday, work it up, and the DNA is usually fine for most applications that we do. The longer it sits, the more likely it's going to be degraded, but typically DNA is much more stable. The only key to DNA, though, is you want to make sure that you avoid the green top heparin tubes. Heparin is a sticky molecule that tends to co-purify with DNA, and that's fine, but it turns out heparin is a really nice inhibitor of Taq polymerase, which is the key enzyme in PCR reactions, and almost all DNA applications require a PCR step. So if you're going to be getting DNA, you really should avoid, or RNA for that matter, you really should avoid the green tops. We typically use purple EDTA tubes for those. Sometimes people like to use the yellow tops, ACDs. Those work fine too. But in our experience, DNA purple tops is the most stable, particularly if you're going to have issues of collection at a remote site and there's going to be shipping involved. They can really sit in that for a couple of days without substantial degradation. RNA you can't do that with. So if it's RNA, you really got to isolate cells immediately and get that material stabilized. And there actually is an example here in UAB where we have helped study isolate RNA from plasma. Again, this was from EDTA plasma. And they were looking for markers of colon cancer, and this was published just last year. Dr. Wilcox was the PI here at UAB working with a group at Hudson Alpha, and they discovered a set of microRNAs, which is a type of RNA that is found in circulation that is predictive of the presence of colon cancer in patients. So it could be a potential biomarker for the diagnosis of people with cancer. But again, a lot of technical details go into that. So those RNAs, those plasmas need to be worked up immediately. So they actually had coordinators in the clinic at TKC. We were right there at the bedside or on the table side getting blood, running it over to the lab. We were spitting it down immediately. When you're doing RNA from plasma, you need to do, it's not a standard centrifugation step. We'll talk about this in a few minutes about centrifugation. You need to make sure you get rid of all the cells. You only want RNA from plasma, but it can be done. And when they're worked up quickly and stabilized, you can get good results. So we talked a little bit about the collection of samples. What I'd like now to move on to some of the other issues related to specimen handling after you collect them. So your samples have to be labeled, and these are actual pictures from the lab. We get sometimes tubes where labels are partially ripped off. You can't really read it, and what the heck is this? It's not good. So you want to make sure that your samples are properly identified and labeled, that they've got the relevant information on them. You need to make sure that the samples are processed within a time frame that is required for your future testing. So again, with RNA, you can't let it sit around. That's just not going to work. With DNA, you've got a little bit more flexibility. You need to make sure that the right vacutator is used, as we've talked about. So remember some of the general rules. If it's DNA, don't use a green top. If it's RNA, you need to work fast. Serum tubes are these orange or tiger top ones. These are the ones with the gel barriers in them. That will give you serums that will clot. Purple and green plasma, which doesn't clot. And then you need to think about how you're going to store them. What type of mechanism you're going to use to actually put these in the freezer and retrieve them. So why is all this important? I hope it should be obvious that the results are only going to be as good as the conditions that the specimens are collected under. And if you don't collect the specimens the right way, you're not going to get the right result or a proper result. Proper handling and processing is necessary to ensure that the right specimen from the right patient is recorded and that information is useful for your research. And again, just without saying that quality specimen management is required for accurate test results and meaningful research. So this really comes back to, as coordinators and implementers of research, you need to know your protocol. You need to know what sensitivities are under specimen that are sensitive to light, to temperature, and time. You need to know when are patients coming in the clinic. If it's Friday afternoon and you want to get RNA from a B cell subset, it might be difficult, right? Because that's going to take many hours after specimen collection in order to get those cells separated out and RNA isolated. So you need to kind of think these things through when you're designing a study. what's going to be the feasibility of getting the blood collected in a way that's going to allow me to get a sample put away in the right way within a timely manner. You need to understand where the specimens are going and what's going to happen to them. So one of the issues then is you need to know how to use the centrifuge. And in our CCTS core lab, we have this issue all the time where people come in and want to use a centrifuge, but they don't really understand how the centrifuge works. On a centrifuge, there's really three issues: temperature, time, and the speed that the sample is spun at. One of the points of confusion all the time in the lab is when you spin the sample, you have G-force versus RPMs. They're not the same. And if somebody says, well, just spinning at 2,000, okay, that doesn't really mean anything. Is that 2,000 RPM or is that 2,000 times G? The times G is the number of times gravity, so that's going to vary with the rotor, with the diameter or radius of the rotor. The bigger the rotor is, the more G-forces you're going to get from that sample as it goes out to the bottom of the tube. Whereas the RPMs might be the same for different rotors, the G-force is going to be completely different. So you need to understand those differences and know what the appropriate settings are for your particular application. So I mentioned the importance of labeling. I would always try to err on the side of using pre-printed labels when possible. I can't tell you how many times we get samples in our core lab where we just can't read it, and we don't know what it is. They don't come with a protocol on it. You know, when it's going to a core lab or even to a clinical lab, there have to be appropriate identifiers on it. We work with over 100 different studies. We need to know what the study is. We need to know what the patient ID is. So if it is handwritten, just make sure it's written legibly. And make sure that it's not written in a marker that's going to smear if it gets wet. Sometimes it's going to put on ice, that label's going to get wet, and then the ink smears, and you can't do anything. So if possible, pre-printed labels are best. For labeling of the aliquot tubes, it's even more important to use pre-printed labels that are appropriate for the storage conditions that you're going to use. Labels in the freezer, if you just use standard Avery labels, they will come off. They will not last. You cannot use Avery labels on specimens in the freezer. There are a lot of companies that will sell freezer labels. You need to know whether you're going in an ultra low, like a minus 70, minus 80 freezer, or whether you're going to cryogenic temperatures, which would be like a minus 150, minus 160. That's typically liquid nitrogen freezers. That's most typically for cells. And there are labels specifically made for these applications, and they don't come off when they're used appropriately. The issue with pre-printed labels here again is if you use an appropriate label that's appropriate for storage conditions, they can be pre-printed in such a way that the ink does not come off. In fact, we use one label where it actually has a clear half where it wraps around and the clear part covers the printed side, so it actually can never be rubbed off as it goes in and out of the box, and is protected, so you can always read what's on it. I would advocate that samples should always be barcoded with a unique aliquot identifier, so every aliquot has a unique identifier. So you might have a given patient, right, who has 10 aliquots of something, but each aliquot will have a unique identifier that will identify that specific aliquot. It will also be identified by the patient ID, so that would be the same on all 10, but each individual aliquot would be different. And it's really important to think these things through because once it gets into the freezer, you cannot relabel it. And I will just say that categorically. People say you can. That's really hard because the tube is wet now and nothing's going to stick with that one. So you've got to have your IDs worked out. You've got to have your labeling scheme worked out. You've got to have your label printing scheme worked out before you start putting things in the freezer. You can't drop a sample off and say, well, I'll get you that later today. That just doesn't work. You've got to have everything lined up when that sample is collected. So everybody in the chain of custody knows what the sample is and how it's supposed to be labeled. So what about freezer options? Typically speaking, in the laboratory, there are 3 different types of freezers. There are minus 20 freezers. Those are kind of like the freezer at home. The difference, though, is in the laboratory, you don't want a freezer that cycles. So those are the frost-free freezers, right? Your freezer at home is constantly warming up and cooling back down, so you don't build up ice. It's really nice. You don't have ice buildup, but that means that your samples, whatever, is going up and down in temperature all the time. For biospecimens, that's not good. So you got to make sure if you buy a minus 20 freezer, it is not cycling. Then we have ultra lows, which is minus 70, 80 range. That's typically where most biobanking occurs for plasma serum. Sometimes people store cells in them. That's not optimal. You can store blood in there. The one point I make about blood, if you freeze blood, when you thought the cells were lice. And so if you're interested in cells, you don't want to freeze blood typically. Now, there's some exceptions to that, but you just don't want to freeze whole blood. They're a way of cryopreserving cells so they can be thawed and used again. And when you think about minus 80 freezers, you can go crazy. There are standard minus 80 freezers, which most of us see in the lab. Then there are really fancy ones, which actually are fully automated systems where everything is barcoded. The freezer can actually go in and you tell it what it wants. It pulls out the specific foods you want, gives them to you, and then you can put them back and say, here's the sample. It scans the barcode and knows where to put it. Those are really nice, but they're really, really expensive, like hundreds of thousands of dollars. This is actually a picture from a colleague of mine at the University of Florida. They invested in one of those fancy machines. It's a nice toy to show off, but they've only got one. It's so expensive. Then the third type of freezer are the cryogenic freezers. There actually are mechanical freezers with compressors that will pull down to cryogenic temperatures. Again, they're usually pretty expensive, and they're not always trouble-free. Most people, when they're storing cells under cryogenic conditions, they use a liquid nitrogen freezer. There's a picture of a typical one here. These can either be manual filled, which means that you put a hose up to it, you just dump in liquid nitrogen every week or more often, or they can be an automatic fill where it's actually tethered to a liquid nitrogen source tank and has a sensor that fills up when the liquid nitrogen goes low. In the CCTS, we have a whole bank of cryogenic freezers that do automated filling with regular liquid nitrogen shipments. The other thing I'd suggest you think about is when you're doing a study or thinking about starting a study and you want to store specimens, do you really need to buy another freezer, or are there other resources on campus that you can use? Freezers, while we're not paying for electricity ourselves directly, they use a lot of electricity. I've heard that the Shelby building is the worst violator of electrical use. We've got a lot of freezers in that building. So really think through, is there a place that I can hook up with to store my samples? I would suggest that the CCTS, we have a very large freezer capacity, unlimited, but nonetheless it is large, and we can accommodate a lot of samples. And so if your needs aren't large and you don't need a whole freezer, why don't you partner with somebody? The other advantage from the CCTS, I'll show you some software solutions that we're using for storing samples, that we have a very rigorous, robust inventory system that can track your samples. So just don't do the knee-jerk buy freezers. Think about, are there ways that I can synergize and work with other groups that maybe already have systems in place to help store my samples. Once you have a freezer, how are you going to store your samples? And here are some horror stories. This one isn't so bad, but typically you open most laboratory freezers with racks and stuff all over the place. Some things are labeled, some things are not. There are boxes, and people know, well, that's so-and-so shelf. I don't know what's in there. Sometimes they look like this, not very useful, and other times they look like that. You really don't know what the heck's going on. What I would argue is you need to get a very good inventory system, and all the vendors sell these. You can buy racks and boxes that go in the rack, and within the box you have these grids, and every single aliquot is in a grid, and a good inventory system will know exactly where every single aliquot is in your freezer. And I'll show you how we do that. So what are some of the issues with ensuring the quality of your specimens? You need to make sure that your freezers are working. So are the freezer logs, are the freezers actually monitored? Now in the CCTS lab, this is all done, they're actually monitored 24/7, but in most research labs, they're not. You could argue, well, you probably don't need to look at your temperature every day, but I'd argue, how valuable are your samples, right? You put a lot of work and time into collecting the samples. You really need to make sure that they're stored correctly. Are your freezers monitored? Are they connected to any other alarm systems? What happens on Saturday when they go down? How are you going to know about that? All the more reason if you're doing a study and you need to start storing samples, there's a lot of expense here, and there are ways to synergize. The CCTS can help you. We have a lot of these things in place already, and we can take over some of these responsibilities for you. They're still your samples. We don't own them. They're under your control. But they're stored in such a way that all these quality control issues are taken care of. You need to make sure that your freezer has a clear list of names to call. And I'm bad about this too. In my own lab, we had a freezer go down and found the guy who was first on the list that left 5 years ago. Very useful. So you need to make sure your call list is up to date. And more currently, people are now going to remote monitoring systems where actually they're monitored through a wireless system where you can use it on your cell phone and check the status of your freezer. Those are pretty expensive, so not most investors when you get that, but that's the way that we're moving now to monitor all our freezers. Is your freezer on backup power? It's very rare that we lose power here, but it happens sometimes. right? All of our freezers in the CCTS are on backup power. We've never had to test that, actually, but nonetheless, we've got confidence at least that if something happens, there's hopefully backup power available. And then there are issues of lab certification that we mentioned at the beginning of the talk, particularly when you're working with samples that are going to be relevant in the clinical domain. What type of certification do you need in the laboratory? CLIA/CAP are typically most commonly found laboratory-based certification processes. Though those typically are oriented towards assays, not towards sample processing. CAP, which is the College of American Pathology, has a new certification for biorepositories that we're actually undergoing now. It's a fairly long process. But you need to think through, so what kind of certification do I need? And then what do the staff need to know? Obviously, at a minimum, the staff need to know appropriate lab techniques, whatever you're doing. But then there's a lot of regulatory issues. And as much as we all like to complain about these things, these are important. Staff need to be trained in bloodborne pathogens. There's an online course that everybody should take. That should just be you should even think about it to do it. General lab safety. Then as you get into more clinical research, there are issues of GCP and GLP, good clinical practice and good laboratory practice. UAB does have a subscription to the GCP module for the CITI program. That's available for anyone to take who's involved in clinical research. We currently don't have a license for the good laboratory practice training. I'm working on that to see if we can get that here. Typically, GLP really only comes into play when the FDA is involved in audits of the facilities that are conducting clinical research. And then there are IATA, which is regulations for shipping specimens. You need to ship samples somewhere to a clinical lab or a core lab or to a collaborator. You just don't throw them in a box and send them. There actually are issues you need to understand, particularly about specimens from patients. Those are potentially infectious materials, and there are rules and regulations on the for shipping those. The UAB has an online training course through the IATA that you can take to ensure that you're following appropriate regulations for shipping. These are all important things. We complain about them, but they really are important. We need to do them. So the last thing I'd like to talk about then in specimen management is how do I manage these specimens? How do I know what they are? How do I know what they are? How do I know what I have? And I think as you think about this, there are certain features I think that are really required that are just you have to have, and other things that are not. So things you have to know. You have to know. You have to be able to track your specimen. When was it collected? Where was it collected? Where did it come from? Again, I would advocate that samples need to be barcoded with every aliquot assigned a unique ID, so you know exactly what that aliquot is, where it came from, and where it went. You need an audit trail on all your specimens, so you know what happened, who did it, what happened to it, and why. It has to have full specimen life cycle tracking. So the CCTS, the system we use, is actually part of a clinical trials management system called Encore, which is now on campus, that allows us to know it's collected in a clinical trial or a clinical study in a certain visit. It comes to us, we can track when we got it and how we stored it and where it went. And then if it gets shipped out, we can track when it got shipped, who it got shipped to, and who shipped it. You need to have reporting functions, obviously, so you can generate reports on what it is you have or don't have. And you need an ability to import information. A lot of times you get a box, here's a box of samples, and here's information about them. You need to be able to get that into your system so you can track what you have. Other options that are good, web-based system is always preferable to something that requires software installation on the OPC. Encore is a fully web-based system. The server actually runs behind the HSX firewall, and we can get into that generally anywhere on campus. You need that ability to create labels fairly easily. Again, that's a labeling issue. You need to be able to lay out the label, so put the information that's relevant. Not every study has the same information that's needed. In a lot of clinical trials that we work on with CCTS, they have a lot of visit information they want on those labels. That's just not relevant to other studies. And you need to be able to actually print those labels. So you need label printers. The type we use actually are called thermal transfer printers. So they actually have a ribbon, and it transfers that information to the label itself. Those are very stable in the freezer. They don't really rub off, and we found that they work very well. You also need to think about having shipping logs and tracking of shipping. A good system will be able to allow you to do all those types of things in a single platform. So what are some of the options? We use Encore, which is actually a clinical trials management system that has a built-in specimen management module. Nothing is perfect. Encore's not perfect, but it works, and it does what we need. It does it pretty well. Like all systems, it has its own way of doing things. You've got to learn that, but once you learn it, it works pretty well. FreezerWorks is a very popular program for doing freezer inventory. I used that for many years before I migrated to Encore. It's very good at what it does. What it doesn't do is really integrate into the clinical environment well. That's what Encore does a big role. Process to integrate directly into clinical trials and everything managed through Encore. Freezer Pro is another commercial program. Open Specimen is one that people are talking a lot about now. Many of you may remember the CA Big project that NCI had a number of years ago that kind of fell flat. But one of the modules that they developed was something called CA Tissue, and that essentially was an inventory management program for biospecimen oriented towards tissue, but it works for other samples too. When NCI kind of decided that CA Big wasn't really wrapped up to what they wanted it to be, they made CA Tissue available. It's now been taken over by a company in India. You can still use it for free, but if you want support and you want customizations, this company will work with you to help you modify the product to satisfy your needs. That's what Open Specimen. And then there are other options, laboratory LIMS or laboratory information management systems. These are typically used in large core labs to manage instruments and data collection of instruments, but many of them have specimen management capabilities too. Obviously, there's cost with this. None of this is free. Probably your least expensive is Open Specimen, followed by these two. Encore is, I'm not sure what pricing is going to be here on campus. It's very expensive for UAB to maintain the license, but how that's going to translate to investigative, I don't know yet. Typically, LIMS systems are very expensive, 6 figures. That's not something that an individual laboratory typically is going to be able to do. If you're really intent on doing something in your lab, I'd advocate one of these. But again, I'd advocate a model where why not go to the people who know what they're doing, and you can do this in a very good system that has good QC and quality control. Everything we do is backed up. It's behind the HSS firewall. Talk to us, and we can help, and sometimes maybe even do your storage for you. Big options that are not good for specimen management are paper records and Excel spreadsheets, or even simple access databases. I've just seen too many examples of where people are well-intentioned, but it's just difficult to manage this way. Particularly paper gets lost. You have personnel changeover, and I don't remember what that was. Excel spreadsheets, they're static, right? So you got to make sure that you have the ability to update. And that's true even in these commercial off-the-shelf systems, right? So once you get information in there, if you pull a sample out of your freezer, you got to make sure you log it out. Because if you say it's still in your freezer, it's not there anymore, then the system shoots this. But that's particularly problematic in Excel or homegrown applications, which is really difficult to build in the kind of give and take that you need for samples going in and out and tracking where they are. So again, if you want to do this, talk to us. We've been doing this for a long time. We have a lot of experience with a number of these systems, and we can help guide you and maybe help you do it if that's appropriate. So I'll give you a couple examples of screenshots of things we do on Encore to show at least how we think about the software side of managing our specimens. And the issues here, when you think about tracking your specimens, you always want to know where they are, how did they get there, they're not there anymore, where did they go. You can see in this particular screenshot here, there are specimen number and there are barcode numbers. In Encore, that barcode is assigned behind the scenes, and you can change it if you want to, but the nice thing about it is people assign every hour clock a unique barcode that's different. Each specimen actually also gets a separate ID. That's the key in Encore. It's the barcode that gets printed on the label. You need to know how much you have, what it is, and what its status is. In this particular case, it's just a screenshot of a bunch of stuff that shipped. Oftentimes, particularly in clinical trials, time is important, right? So you need to know the time of everything. When was it collected? When was it received in the lab? When did it go in the centrifuge? When did it go in the freezer? Those are all important things, and so we can build forms that allow us to track all those times and any other information that needs to be associated with that sample. In most research studies, you don't have this level of detail on your specimen, but sometimes this is important to know. And you need a way of tracking this that's consistent and allows you to link it back to your actual specimen. Here's another screenshot. This is one of the things you need to be able to do is to easily view a storage configuration. And we've actually had it happen. You pull out a tray of samples and the box falls and all the tubes go flying all over the place. Oh my God, how do I do it? So you need to be able to go in and say, well, what's supposed to be in grid 1, 2, 3, 4, 5? And you can see here, it's a little hard to read, but this is basically a concatenation of the storage location. So there's a room, a freezer, the rack that it's in, the box that it's in, and the grid location within that box. So it goes down to a specific aliquot, that individual grid location, tells you everything about it. So an Encore sequence number is just the sample ID, so that's the patient ID assigned in the study. Case number is usually the protocol number concatenated with the sequence number and the date. But then there's a specimen number and barcode. Again, these are all unique for each specimen, the specimen type, and the date that it was collected. So it's all viewable in a very nice, simple way. This is just a few clicks and you get a whole box showing on the screen and print it out if you need to. Another type of view that's useful. So what this shows us on a particular participant, there were 3 different encounters, and these are the specimens I collected on each of the 3 different encounters. And it shows you nicely what the status is. These are available, these were shipped, and these are available down here. Sometimes you can use the concept of via session, so they're making derivatives. So typically in our case, blood will be the original sample, that then gets via session, that derivatives are made, we make cells, we make plasma serum, whatever. Those are then in the specimens that are available or shipped. But the system's flexible that we can give it different statuses depending on where the sample is in its life cycle. If you drill down a little bit more, this is taking one particular encounter, and in Encore, in this case, the lab is blinded to the name of the patient, so you can't see it there, but it's behind the scenes. So for study staff, if they want to go in and see it, they can know exactly who that belongs to. But you can collect information about the specimen. Here's the collection time, the preparation time. If it's actually a tissue, there's a lot of information here you can store about the tissue related to that particular sample. The specimen type is here. Again, this barcode, that's a unique number. You can have annotations, very flexible. These are totally customized. We have received in lab as one of the annotations here. But again, we can do anything we want here. And then you can drill down further on an individual specimen. It replicates this information up here, and then the specimen can have other information associated with that individual aliquot. So it's a very flexible system. I think if you're interested in looking at this more, we're happy to show you. If you're going to manage your own samples, you just need to think about these things. How much detail do you really need to have about your sample? And what's the best solution to store that information? So what about data? So everything I talked about now is you get a sample, you need to work it up right, you need to get it labeled, you need to get it put away, you need to have an inventory system that allows you to track where everything is. What about associating data with it? Well, certainly I've shown you that in Encore we can record information about the specimen, like the time, the date, the locations, who worked on it. That's an audit trail. That's pretty straightforward. How do you associate research results with potentially clinical data? One of the things the CCTS is very actively pursuing is how does one link the availability of specimens to clinically relevant information? Not proposing that that would all be in one place. Oftentimes you have separate databases. You have an inventory database and then a clinical data database, but you need some way of linking. For most research trials, that would be the medical record number, but it could be something else depending on how your study is set up. But you need to think through, what is it I want to know? So a lot of information can be derived from the medical record, and there actually are tools now available that can query the medical record. There's a tool called I2A2, which the CCTS is now using. That is a query tool that allows you to go and actually query clinically relevant information about a participant in the study. But sometimes you have case report forms that are unique. It's not information that's really in the medical record. But there are things that are very important to your study. Sometimes those, if it's using Encore, those can be in Encore. Encore can have case report forms, and you can have your data directly recorded in Encore that would then be directly linked to the specimen, or it could be in an outside database like RedCap or something else. Again, we use a linkage that could be the medical record number or the study ID, and then allows you to link the information in different databases. Just need to think about how you're keeping your information and what those linkages are so you maintain the best use of your samples. So if you want more information that's on this slide here, you're welcome to call me, or I have very good staff, Felicia, Lauren, and Chandler. They're always available to talk. We're happy to show you anything that we do. I'm not saying we have to do all your work for you. You're welcome to do your stuff too, but you can certainly use this as a resource and to talk things through. I've got some web links here that might be of interest. This one in blue here is Edberg, that's the International Society for Biological and Environmental Repositories. We know what the society for that there is. And their whole purpose in life is to think about biorepositories and how they're collected and stored and retained. There's a lot of useful information about best practices. They actually have a set of best practices that we follow now for the handling, processing, and storage of specimens. This is a commercial website, Limco Lab, but it's really nice because it talks about sample handling. Right? So it talks about all different types of blood tubes that are out there and why you might want to use them in different applications. It's just a really nice, simple, approachable website that you can search for different ways of handling your samples. Here's IATA, that's the International Association of Transportation, something like that. It's the shipping people that have guidelines for shipping. And then certainly through UAB, there are a number of training opportunities for different regulatory aspects. And the CCTS website is the last plug for the CCTS, certainly through research commons. There are a lot of resources available, not just our lab, but the clinical research unit, the clinical research support program, which sponsors this talk. A lot of resources available. If nothing else, we're here to help and guide, and certainly help you do your work too, if that's appropriate. But if nothing else, we're here to help you think through what it is you need to do. Then let's not go really.
So two fluids that you didn't mention, spinal fluid and saliva. Saliva for DNA and spinal fluid for peptide analysis.
Right.
But would they go through SPAN similarly?
Yes, we have worked up. We do spinal fluid for a lot of clinical trials. Those are typically, we're given everything we need. We just do exactly. It's usually registry studies and send those out. I think we need to go. I don't think we've done much with CSF for research studies, but certainly we have that capability. Saliva, we have worked a little bit with saliva for DNA extraction. And we've done some buccal swabs too. And we've done one study where when you do a buccal swab, you really have to scrub hard. And that they were very gentle in that study, and we got very little DNA. We just didn't do it right. So again, it all goes back to sample collection. How was that sample? But certainly we can help advise on that. We've not done a lot of it, but we have done some. And I think it's Genotech, I think is the company that has DNA collection kits for saliva. And some buckles. Those are always options. I'm really, if I'm about DNA, I like to do blood. You just get so much more. It's always a uniform quality. It just works, always works. My experience with saliva has been mixed, but it can work.
Other questions?
You said like you were working on the UAB has a GCP part, or you are working on a GLP. It's much more on the clinical side or preclinical side, or it's
That's mostly on the clinical side for clinical trials. So what we're finding is that for industry-sponsored drug studies, that they're starting to ask more and more the people who are handling these samples, are they trained in this, and are these certifications in place to demonstrate that there's an environment that these samples were handled in an appropriate way, so they can trust with the job. And for most research applications, that formal training is not necessary. But I would argue that I think it's still important because it sets a mindset. How do you think about this? It's not just I just go to the lab and just do whatever. You need to understand what you're doing.
I would add to that with industry trials, we do HIV trials, and all of our sponsors are looking for all these different trainings. They want your lab to have everything you described. So thank you. Any other questions? If not, that was a very interesting, comprehensive presentation. I learned a lot. I'm a clinician, and I always wonder how why do I have to have this exact tube? And why do they care how long it takes to spin? It was great. Thank you.
Yeah, it's, I don't know, 104 years ago. We got specimen.
No, it's like you can research and you have straight computer, you can research for your specimens. Since it's all in that database. I'll let you see. Ask me a question. Thanks again.
Yeah, well, just quick.
Yes, but, so, with the CCTS units, are all specimens that get collected by individual investigators for research studies. Right. So, I can't tell you.
No, I agree.
I can give you 52.
No.
I can tell you who has them.
Yeah, that's what I did. And I will reach a bit because I know because I work in the company, I know that it has to post lots of paperwork, and then I can tell that I cannot go there. So if they will draft it, I will email. That's the point. Okay. Yeah.
So, like, if you know you want.
Yeah, that's why it's that's right.
It's like 80 we have.
Yeah.
So, it's not for me to say.
Yeah, right, I agree.
Other than that.
Yes. Yes, exactly. All right. Thank you.
Certainly things that we have a lot of all materials on the radio.
Right.
Highly collaborative our consent form.
Yeah,
Written go out.
the numbers we've got before.
That name is
Healthy.
Should I know him?
Maybe
that's why his name is from.
Oh, that's no, we don't need that. We're talking about numbers, saying less than 10 numbers. Yeah, just to get more. Yeah.
We've done that with all of other.
Yeah. I'll let you go on. I'll email you. Thanks.
Sorry about bone marrow. Some differences.
Yeah, so, we haven't worked on any bone marrow samples, my knowledge. Well, again, the same principle.
It's like
Issue there is
No, I mean, I don't.
Bits and spurts. And with cells, when we do PBMC, which is not we usually crowd preserve them, so investigators. Because they want to bank them all together and then do their assay.
Well, it's conferences.
Yeah,
This trip was
another
a
institution.
site visit of the accreditation of another institution.
Get the sample of black coverage and
Work
cell layer.
very intentional. It's my first time.
Not lot. It's going to do a total DNA extraction. Freeze all your cells. So that whole process, is that something you guys could do?
We do that all the
We
time.
have our own at we
And
have
yeah.
So, in theory, bone marrow, it's just a variation.
the arbitration is essentially accreditation of human research.
I want to touch it for you guys, bosses. If you're
And
elected
so our accreditation is
And do you do that where after you
just human,
sort of like
human research.
you've got your money through cell layers, you do cell counts, you can figure out how many cell plots to create?
Yeah, so it really depends, so we do with study. So, there's one study that says, he's getting 2 grade
We have a good
and he
program.
just wants 4 outcomes.
And these can be documented.
Another study we actually count, so we've got one of those, Victor has something called
All the key players.
automated counter.
Okay, so you
Basically,
have-
hemostatometer.
I was gonna say, we use it.
Yeah, it's a fancy hemostatometer, you get a little machine and you display, and has a camera and it counts.
Okay.
We
Actually,
used to send people out of.
We have a full-action microscope.
And then the last thing is, I'm in the process of,
We've got a project.
Well, I used to, I mean, whenever,
and how does that all work?
occupational health and safety.
CCTS support versus reimbursing you all.
I could
for
show.
that.
Yeah, so,
I
that's kind
send
of
people out to get out of here.
changing
You don't
landscape.
work here.
So the CCTS program in the past.
Yeah,
able to
I'd be like,
subsidize
they're coming
the
tomorrow, please don't come.
The NCAS, because the actions to
Yeah,
response to
don't,
CCTS.
no, go away.
There are a lot of,
Well, I mean, because these
they're not
are,
interested in it anymore,
Oh,
and I
yeah.
think that the other institutes
Oh,
that fund the research should be paying
just,
more
I
cost
mean, as soon as you start
of the
asking
work
what they
that
were doing,
is done.
they, well, you know that they do this,
So,
and they do this.
we have a price
This, this,
sheet,
well,
that we change it.
oh,
It doesn't change the cost to do the research,
yes,
it's just that we used to get money
You know,
from CCTS
and,
people. Now I've got to float them more from user
but,
fees.
yeah.
CCTS
now I
will
feel
still
like just
direct
go away.
But it's, you know, contract, what is that?
Do a PV report.
They already have the different thing. So they just.
The cost of my call and
People out on call.
Do you want to send
trial
these
of
people
months.
troublemakers to us, and we can show them what Dr. Garrity does?
But, you know, certainly if you want to talk more on contract.
I had a meeting with, OSD.
How we price things out,
And
and see what it would cost.
They were
And
like,
in terms of scale.
and I was
because she
saying,
has
Dr. Garrity wants
functioning
to
at some
change,
markets
and I told him that I did it.
pediatric
And they were
rare,
like,
like,
after you told her that.
communications will get like 40. But there could be.
She's so
multiple
excited.
times that, you know, do
To think
those
that people
things. So it's not
actually
gonna be
think
like-
that she's their employees.
But, my other question was, for me, I'm trying to get together the clinical database side of things, so we're using something like that would be okay, database as long as we have, you know, a linker, which we
No,
use.
she's bigger work with.
And so when we store things, we can do it as a medical record number.
I like that,
Sometimes we don't have another.
I like that I can argue with her.
Number.
that she's not one of those people that's like, no,
And
this
it's
is
got
what
different
this
structure,
is
but
on me, and
it's a 99 dash, it's really good. So we can have, I don't have medical records, but we'll still have those.
Yeah, all of ours, whatever, you get
You know, like I say, I'm more behind the side, so I want to do that.
Yeah, that's some of the things, so when I actually looked at the database, so,
Because then I'm sorry, but.
So what Encore does with the medical record, because it looks, when it looks back to the UAV, not more than the shortest.
yeah.
It's like this, I was gonna be like,
No, we have a
regulations.
regulation action.
Some of them will, because we actually do our flow of UAB and not assign that for a so a lot of those looking good would have a UAB. And we actually track that in the right campus and show us and UAB tracking numbers, but just depends on the next generation.
we,
Yeah. So,
she does, she teaches you how to think.
Well,
I was
I mean, that's, it's not
like,
just.
I'll be right back, 2 hours. I just want you to know I was channeling.
But yeah, I mean, you know, 40 a year.
So I don't think so.
I mean, we go through 34,000.